James, please stop.
Hmm?
That's talking about him. All right. A little early because I didn't want James to go all the way down the entire minute. The calculator. You need to, there's a database question at the beginning. Actually, I'll wait till 8 o'clock in case other people are waiting. I was writing down what's on the test. Okay, there's a database question, like there's a graph and you have to interpret the data from it, and that's what you need to calculate for. So you have to crunch some numbers or figure out percentages, or it's, you may not need a calculator because it's not hard numbers, but you can have one just in case. So there's like a graph question at the beginning that you can't study for. It's kind of, you got to be able to interpret the graph, and you may have a calculator for that, and you may use it. Let me just go through some things. You have to be able to transcribe DNA to RNA to mRNA. What molecules are used in transcription, like DNA, RNA polymerase, RNA nucleotides, things like that. The functions of lactase, how it's used, the translation process. That likes me a good process. The different structural levels of proteins. I'm not going to tell you which one because it could vary from class to class. You have to know functions of proteins. There's 3 of them. They may vary from class to class. You have to be able to draw a dipeptide, and label the bond, the peptide bond. Large ribosomal subunit. The ribosome is 2 subunits. The large one comes in after the first tRNA binds. It's just part of the ribosome. No, it's just part of the ribosome. You have to know its purpose. The purpose is just to be part of the ribosome for translation. It comes in after the first tRNA. I don't think you need to worry about the sites and all that stuff. Methylation. There's some extra credit questions, so I'm not going to say if what I'm talking about is what's going to be on the test. If it's not what I'm talking about, it may still be a multiple extra credit question. But methylation is not on the main test. The six specific ones, the protein functions of the 6 specific ones. Methylation may be extra credit question. We shall see. What else was there? How to measure photosynthesis. There's 3 ways. There's 2 that are direct, one that is indirect. Explaining how they're measured would also probably be beneficial instead of just saying, input of CO2, output of oxygen, things like that. Explain how you measure it. So dipeptide, peptide bond, functions, the structural levels of proteins. One of the photosynthesis processes will be on the test, and again, it may vary from class to class, but you should know one of them will be on there, but not both for any one test. Origins of oxygen, how enzymes are affected by pH and temperature. And that is it. I just told you everything's on the test. Any questions? I know you're looking at this and be like, well, that's everything. And it kind of is, but it's not all of everything. It's a little of everything. Except for methylation, evidently. Any questions about anything you may not understand? Well, I'll go through it again since we have some time. There's a database question that you can't study for, but you will be able to use a calculator for. Be able to transcribe DNA to RNA. The molecules are used in transcription, like DNA, RNA polymerase, RNA nucleotides, stuff like that. Lactase, how to measure photosynthesis. There's 3 ways, 2 direct and 1 indirect, and how they're measured. The process of translation, how pH and temperature affect enzymes. One of the photosynthesis processes will be on each test, may not be the same one though. Origins of oxygen, how to draw a dipeptide and label a peptide bond. Functions of proteins, there will be 3 of them, again, may not be the same. And structure of proteins, which also, again, may not be the same. You have to draw R groups for specific examples. You can just put an R, that's fine. When you say the process of photosynthesis, you mean light dependent? Yes, the 2 processes are light dependent, light independent. One of them will be on each test. May not be the same one though. We'd be asked about immobilized enzymes. I said that you should know the lactase thing, which is an example of that. Representing the visible light spectrum, do we have to also draw the carotene? No, you don't have to draw either of those. If you were to do it, I would know chlorophyll A and chlorophyll B, but it's not on here. Do we need to know the photosynthesis squiggly graph? I just said that. Can you explain non-competitive inhibition? Non-competitive inhibition is when a molecule binds to an enzyme on a place that's not the active site. It's called allosteric site. It changes the shape of the active site so that the substrate cannot bind, and it's reversible. An example of that is threonine as the original substrate and isoleucine as the end product. Could say the last half of all that again, but a little slower so I can write it down. Do you still need me to? Okay. Is that going to be on tests? I don't know what you're asking about. Inhibition. Again, I'm not. If I didn't tell you, it might be an extra credit question. If it wasn't one of the things I told you to specifically know, it could be an extra credit question. That's why it's extra credit. Know about Calvin's experiment? No. Anything else? 41 people watching. Let's see who they are. How have you been? I've been fine, thank you. I am famous. I have 25 subscribers. I've just found out. I'm on my way. Won't be in class much longer. Be famous. A lot of fans. Yeah. My day was fine. Thank you for taking the time. No problem. Thanks for doing the live stream. No problem. Oh, that's sweet. Yeah. I appreciate it. Does anybody have any questions? Are we done? Yes, monetize. Thank you, James. Work on that because I don't know how to do that stuff. Figure out a way for me to get paid. Functions of proteins, we got to be specific or list or it. What? Why are you on this? You're probably across the table. Anna's actually in her room watching it like everyone else. Thanks, thank you. I understand what you mean about the functions. Yes, you have to know the levels of protein structure. Let's move 5 to 3 for yes. They move 5 to 3. Everything's 5 to 3. You have to know the 20 amino acids. Exactly how much NA. No. You don't have to know the numbers for NADPH or ATP. And nucleosomes help regulate transcription. They kind of sag off the histones and allow enzymes in there to transcribe and stuff like that. What about translation? What about it? It's on there. 3 to 5 is cursed. I don't know what you mean about translation. I said you need to know the process, and I'm going to stand by that. Could you go over how to measure photosynthesis? Sure. Directly, the amount of CO2 taken up, which can be measured with, if it's an aquatic plant, the pH, because it'll become more alkaline as CO2 is taken out. And with oxygen, you can measure the bubbles that are released. Indirectly, you have to measure the change or increase in biomass. And if you do the ones with oxygen CO2, it helps to say like in a container over a set amount of time, just to make it more lab-esque. My wife's watching Moana.
What?
That's telling me you're watching Moana.
An excellent movie. They should all try and.
I have not seen The Joker. We do need to know how many. You do need to know the origins of oxygen. I'll just leave it at that. It's not Diet Coke, it's tea, actually. Make way. All right. Well, if you guys have any more questions, do we have to know the types of bonds and tertiary structures? You need to know the levels of protein structure. That is all I will say. They appreciate you, Cheryl. They appreciate you.
Why?
I don't know, Megan says that they do.
Thank you.
Could you briefly explain Calvin cycle? CO2 is fixed to Ruby P by Rubisco, which is an enzyme, and it makes glycerate 3-phosphate. Glycerate 3-phosphate gets reduced to triose phosphate with NADPH and ATP, which was from the light-dependent reaction. Some of the triose phosphates are used to make organic molecules for the plant to use or store, and the remaining ones are regenerated to Ruby P with the help of ATP. That's the extent to which you need to know that. If you include that the NADPH and ATP came from light dependent reaction, I think they give you credit for that or something. For the test, do we have to put glycerate 3-phosphate or G3P? I've heard glycerate 3-phosphate because there's other molecules that are generically called G3P. So just to be safe, you have to explain end product inhibition. That is the same as non-competitive inhibition, which, as I said, is not among the things I said you need to study, but there's also extra credit, which may or may not include that. I don't remember what the extra credit was. I'm not gonna lie. Actually, I only made one extra credit. I got to do 2 more. So maybe I'll make that one of them. I don't know. Wait, so it always goes light dependent, light independent, like one to the next. I understand what you mean. Once the NADPH and ATP and CO2 are available, then it goes through like independent reaction. It's not like it's not so much a linear thing. It's more of a this is happening, and then the products are then used for this to happen whenever. Because sometimes some plants that can store the CO2 that they take in at night, and then during the day when they have light, then they could go through photosynthesis. So it depends on what type of plant it is. Because yes, I think I got that. Do we need to know how to draw the graph? No. Okay. Does the pH drop or increase after photosynthesis for measuring CO2? Increases. Yeah. James dropping his chemistry knowledge. Indeed. When we talk about the respiratory system, we talk more about that just for fun. So the only thing we need to draw is dipeptide, correct? I am comfortable saying that if you could draw a dipeptide, you can answer the question. Yes. You might get mad at me. Oh well. Do we draw simple? Just the dipeptide. If they ask you about a condensation reaction, then you'd have to draw that stuff. You sure you know how to draw an amino acid, so it should be hard. If anybody's interested, Mr. Perry's killed me in fantasy football. Disagree. All right. If there's no more questions, then I'm going to end this. One last time. Any other questions? Wait a minute. Is it? He had Stephon Diggs. Will you pass? I believe so. Cyclic fluid phosphorylation. No. Needn't to worry. Are they the same? It's all the same until it gets to the end, and the electron goes back to electron transport chain instead of reducing NADP. Then it goes back to the electron transport chain, pumps some more hydrogen ions to make more ATP before it reduces NADP. Mwahaha. That it? Good. All right, I'll see you tomorrow. Best of luck. Get some sleep.